So with problem 1017, we want to take a look at how do covalent bonds differ from hydrogen bonds, and then we need to define base complementarity. So I think since this is a genetics course, let's take a look at let's look at some nucleotides, right? So we want to memorize these anyways. So let's start drawing. I think adenine. Okay, let's do adenine. So, okay, looking good so far, I think. Let's see. Okay, this is going to be our adenine. I'm going to draw a nitrogen up here, two hydrogens. We're going to have a double bond here, a hydrogen here, a double bond here, and I think that is maybe a double bond here also. Yeah. Okay, so I think that looks pretty good. Okay, so I also want to draw thymine. So I want to draw this thymine base paired by hydrogen bonding with this adenine. So draw it like this, and then we have to draw it in a kind of reverse orientation from how we're used to seeing it. Sort of flipped around like a mirror image almost. So thymine, this is going to have an oxygen here with a double bond and a methyl group here. We're going to have some double bonds here. Let me see. One is going to be 1, 2, 3. I think it's going to be here, and the other one is, I think that's it. And we're going to have a hydrogen here. Okay, so we have adenine here, thymine here. We'll put a hydrogen here. These would be the bases, the nitrogens that are attached to the 5-carbon sugars in the nucleotide. So why don't we write that instead? Get rid of those hydrogens. Write sugar. Okay, so what is a covalent bond? So a covalent bond would be any of these bonds I'm showing here, any of these lines that I've drawn here. Those are covalent bonds, and what's going on there is these atoms are sharing electrons. So they're actually sharing the electrons, and that results in a very strong bond, a strong bond that we call a covalent bond. Okay, so that's what we need to know about covalent bonds. So those are very hard bonds to break. So the hydrogen bonds form between these base pairs, and they're much weaker bonds. So what will I do here? I want to erase that so I can show you kind of what the hydrogen bonds would look like here. So this hydrogen here will hydrogen bond with this oxygen over here. And so hydrogen bond kind of, how does it work? I think the theory is essentially that this nitrogen here kind of pulls away the electron, takes more of the electron that they're sharing from hydrogen here. And we end up with a slight positive charge here. And this oxygen here is a slight negative charge. And because this is positive and this is negative, these guys are attracted to each other. Again, so they're not sharing an electron like in a covalent bond, so this bond is weak and it's easy to break. So here this nitrogen has a slight negative charge, and this hydrogen here has a slight positive charge. And so we get a hydrogen bond forming here. So these two hydrogen bonds are the hydrogen bonds that form between adenine and thymine. Okay, so that's the covalent bond down here. Again, they're sharing an electron, and these hydrogen bonds forming up here, they're just sort of sharing a charge. They're just attracted to one another because of these charge differences. So, and the next one is of problem 1017 is define base complementarity. So when we're talking about base complementarity, so we're just, what that means is with the DNA, so if I give you a DNA strand, one strand, starting at the 5' end, going to the 3' end, you know that this A is going to pair with T, C is going to pair with G, G with C, and T with A. So just by having one strand, and because these bases kind of complement with this strand, we can determine the sequence of the other one. So this is the complementary strand to this strand up here. So base complementarity means for every given sequence of DNA, you can come up with a complement of it. And we can come up with that complement just because we know A pairs with T and G pairs with C. And I can draw the hydrogen bonds in here. So 2 bonds with A and T, 3 bonds with G and C.